In this video, we'll talk about the process in which DNA is copied, and also talk about what happens when there are mistakes or mutations. So let's talk about the process of DNA replication. In order for double-stranded DNA to be copied, the first thing that needs to happen is that the two strands need to separate from each other. This occurs at a specific location known as the origin of replication. So, in this replication process, first hydrogen bonds between the two strands must be broken. Those two strands of DNA then unwind and separate. Each strand can act as a template for the other, following the DNA base pairing rules. So if you have an A on one strand, you know the other strand should have a T in that location. If you have a C on one strand, you know the other strand should have a G at that location. So new bases are now being added to a growing nucleotide chain that's complementary to the template. And this growing chain is following the base pairing rules. The result of this is that while we had started with one double-stranded molecule of DNA, we are now ending with two double-stranded molecules of DNA, each original strand acting as a template for the newly synthesized strand. In this way, we end up with two identical copies of that double-stranded molecule of DNA. Now, one thing to keep note of is that in these new DNA molecules, one of the strands was from the original, it is the template, whereas the other strand is newly produced. We call this type of DNA replication semi-conservative replication. Because one strand of the original molecule is conserved in each of the new double-stranded molecules of DNA, although one strand is also newly synthesized, so it is semi-conservative. So again, in this process, we start with double-stranded DNA. The strands separate by the breaking of hydrogen bonds. Each strand now becomes available to act as a template for the enzyme which is copying the DNA. And so we get newly synthesized strands, which are complementary to the DNA template. The enzyme responsible for this copying of DNA is known as DNA polymerase. Now, enzymes typically end in the letters ASE, and usually the name of the enzyme tells us what it is doing. And so DNA polymerase, as its name suggests, it is making polymers of DNA. It is the main enzyme involved in DNA replication. It adds the nucleotides with bases complementary to the template to the new DNA strand. The location in which this replication starts is known as the origin of replication. This is the location in the double-stranded DNA where the two strands begin to separate. For prokaryotic chromosomes, there is going to be a single origin of replication, whereas eukaryotic chromosomes may have several hundred on a single chromosome. DNA polymerase is the enzyme that's responsible for copying the DNA. And it does a really good job. In fact, if you could type at 50 letters per second and make a mistake once in every 10 million bases, you would consider yourself a pretty good typist. Well, that's what DNA polymerase is able to do. Yet with that said, DNA polymerase does occasionally make a mistake. We call this type of mistake where it puts in the incorrect letter or a non-matching base. We call that a mismatch error. Now, DNA polymerase, it does have some proofreading activity itself, and we do have other proofreading enzymes within our cells, and they help us to correct these mistakes. One mistake every 10 million bases doesn't sound like a very high error rate, but when we consider that we have 3 billion bases in each one of our cells, we would be looking at several hundred mutations each time a cell tried to divide. And that mutation rate is just too high for our continued survival. We call these changes in the DNA sequence mutations. So one source of mutations could be these incorrect base pairings that are put in but are not fixed. Another cause of mutations would be changes to the DNA caused by mutagens. Now, mutagens are physical or chemical components that interfere with the copying of DNA and can incorporate some of these mistakes. Now, there's a certain window in time in order for these mistakes to be fixed. However, if they are not fixed before the next round of replication, then these mistakes could become permanent. Mutations are the source of new alleles. Sometimes these mutations will be harmful, and that's the case with genetic diseases and disorders. Occasionally, these mutations could be beneficial. An example of this would be a bacteria which had been sensitive to antibiotics, now no longer being sensitive to that antibiotic because of a mutation. That's an example of a beneficial mutation. But it turns out that most of these mutations are actually neutral, meaning they aren't specifically harmful, nor are they particularly beneficial. What I'd like to do is show you a few examples of human genetic diseases or disorders that are caused by changes in DNA sequence. One example is the gene that causes cystic fibrosis. Cystic fibrosis is a recessive genetic disorder, meaning if an individual has 2 copies of the disease allele, then they will show that disease. And really what causes the disease allele to be different from the normal allele is a loss of 3 letters, 3 nucleotides. That's the only difference here. Now we can also look at the difference between the normal allele and the Huntington's disease allele. And in this case, Huntington's disease is a dominant disorder, not a recessive one. It takes just 1 copy of this dominant allele in order for those individuals to show these traits. In the normal gene, there's a section of GTC repeats that is repeated anywhere from 9 to 35 times normally. However, if those number of repeats increase from the 38 all the way up to 250 range, this can end up resulting in a protein that can cause the symptoms of Huntington's disease. So what does our cell do if it is able to recognize damage to DNA? Well, DNA repair is something that a specific group of proteins within our cells undertakes. And this DNA repair, it consists of 3 steps. We call these the 3 R's of DNA repair. So we have recognize, where the damage on the DNA strand is identified by a specific pair of protein and it notifies the cell, we have damage in this location. Then a different enzyme is going to come and actually cut the DNA on either side of the damage, and it will remove that section of the DNA. Finally, DNA polymerase will come and will fill in that missing DNA using the remaining strand as a template. So it will replace that damaged DNA. So these are the 3 R's of DNA repair: recognize, remove, and replace. Now these 3 steps to DNA repair are only going to be effective if at least one of the DNA strands is still intact and without damage. And so it is possible to be exposed to high levels of UV radiation for long periods of time, which will result in extensive DNA damage. DNA damage that may not be easily repaired. In fact, some of this damage, again, it may be irreparable. And so the result is that if a cell recognizes that it has extensive DNA damage that it cannot fix, that cell will typically trigger a self-destruct mechanism known as apoptosis. And you might think, wow, that's kind of overreacting. Why would the cell destroy itself if there's just a few mutations it can't fix? However, if apoptosis fails and those mutations are in the genes that control the cell cycle, it could result in that cell becoming cancerous. And so really, it's only those cells that are mutated that fail to perform apoptosis that can end up forming cancer cells in an organism. Now, the last topic I'd like to discuss in this video is a specific genetic disorder in which the DNA repair mechanisms become non-functional. This is a condition known as xeroderma pigmentosa. It is a recessive genetic disorder, meaning that it requires an individual to have 2 copies of that recessive allele in order to have this disease. But in this case, those alleles end up producing a non-functional DNA repair proteins. And so these individuals are not able to do those simple steps of recognize, remove, and repair the DNA damage. For these individuals, even very brief exposure to ultraviolet light will result in severe blistering. These individuals will also be highly susceptible to skin cancer as well. Often, ultraviolet light from the sun will cause thymines, which are residing next to each other, to form a covalent bond with each other instead of the usual hydrogen bonds with the other strand. This ends up being DNA damage that cannot be copied and interferes with the use of that DNA to make proteins. And so, if it cannot be repaired, often those cells will perform apoptosis, or again, they may possibly become cancerous. So with that, that's the end of our discussion of copying DNA. In our next videos, we're actually going to look at how DNA functions when it comes to the production of proteins. See you in the next video.